I'm Ryan, and 4 years ago, I started a bioinformatics and scientific programming core at NICHD and NIH. And today I want to share with you some of the lessons I learned from starting this core that I did not know ahead of time. Of course, this is N of 1. I've only done this once so far, so your mileage may vary. I realized early on that a top-down approach with a core head kind of helping out each core member individually really did not scale well, and I was kind of the weak link or the single point of failure. I realized that really my job was to lower the activation energy so that core members could help each other easily. And that's what these ideas today are about. So the first lesson learned is about documentation. And this is the current state I'm describing. I'm not saying this is the right way, it's just where we are now, how I'm thinking about it now. There's lots of room for improvement. The first kind of documentation is a README. We create this at the start of a project. It has things like what are the biological questions, what's the strategy, why are we doing this in the first place, to provide that kind of context. The audience is ourselves in the future after we've not worked on the project for a while and have to remember what it's all about. The second kind of documentation is what we call a guide, or it's an HTML file, and we ship this HTML file with results to our collaborators. So this is updated each time we ship, in contrast to the README, which mostly is only updated once. The guide has links to the data, has explanation, interpretation, a change log, and when we meet with our collaborators in project meetings, it kind of serves as an anchor point for those discussions. The third kind of documentation is Git. So we make Git commits of the code and the config, not the data. Helps us figure out who wrote what code and when. And one thing we started doing is making sure we have Git tags for each time we ship the data. And it helps us figure out what changed in the code and config between now and the last time collaborators saw the results. Helps us figure out what changed. And this is, of course, updated frequently. So the fourth kind of documentation is what we call a work log. This is a stream of consciousness, informal notes. It has troubleshooting, lessons learned, data shuffling, what to work on tomorrow. We update this as frequently as possible, and it includes everything else that is not code and config. And of course, it is the document itself is also committed to Git. Shifting gears a little bit, the next thing I learned was the usefulness of a consistent directory structure. It kind of doesn't matter what that structure is as long as it's consistent. Why? Because it helps everyone feel at home in every project. It's much easier to help someone when you know where everything is. As a specific example of that, here's a kind of structure that we tend to use with a lab name and a project name that map to GitLab. And importantly, we have a conda environment at the top level. This lets us immediately jump in, activate that conda environment, and use all of the software that the last person was using in this directory. And of course, we have our documentation here as well, and the rest of our directory structure. What I didn't realize until we had enough of these directories laying around is that you can start to do kind of meta-analyses and look across all of the projects to see if there are trends or if things are changing, or even patch bugs and figure out where those bugs might be. And it's only possible when you have a consistent directory structure. Shifting gears again, one of the things I didn't realize would be useful is a utilities package. Once we built this and started using it, it just grew and grew and grew. And at this point, we have things like onboarding pre-flight checks to check permissions and keys, generate templates for the documentation I showed you earlier, maintenance tasks, data management tasks, automation, reporting, accountability. All these things, having a place to put all that utility code ended up being surprisingly useful. So the last thing I want to talk about is scaling. How can we be more efficient in the face of increasing bioinformatics support demand without our budgets similarly increasing? One way of doing that is this hub and spoke model I've been using. And the idea is that we have these embedded bioinformaticians who are providing local support to a small number of labs. They'll specialize in the methods used by those labs, they'll attend lab meetings, they'll just in general build really strong relationships. But so that they're not kind of working in isolation, we also have the central core, which is supporting them. We're building infrastructure, handing off code to them, and also mediating disputes and providing more firepower when needed, and in general just having their backs, so that collectively it's all the core, but we have the central and embedded components of it. When I first started, one of the ideas I had was to provide formal broad spectrum training. That is, teach everyone R and Python, and that would help expand bioinformatics capacity. Turns out that doesn't scale very well. It takes a lot of time. And of course, the people learning this, if they don't practice, it doesn't stick. And it ends up being a waste of time for everyone. So instead, what I've been doing is identifying people in labs who have discovered that they're good at programming or that they have a knack for this kind of thing. And find those people and bring them into the community we've been building with, strong communications channels and relationships to make sure that they're comfortable asking us questions in the core, and that there's this open dialogue. And as a result, we form kind of this auxiliary core of folks who are kind of offloading some of the work we would otherwise be doing, freeing us up for other things. While at the same time, the labs are gaining some local expertise. Often there's much more biological know-how in this auxiliary course. They can more directly address some of their biological questions. The labs gain some level of independence. And I make it clear that we're always here as a backup. So if anything goes wrong, they have a safety net and we'll back them up. So today I've talked about docs, directories, utilities, and some different ways of scaling like hub and spoke and auxiliary core that I hope that will be useful for you in your own course.